Maybe I should issue a warning. When I was bouncing my talk by Ronnie Avisar, he said, oh, we incorporated all of this into our models in 1992. So that's about 20 years ago, and from what I've seen so far, you might have. When I bounced it by Gabi, he said that he will have to request some extra time to fix all the misconceptions and misunderstanding that I will see among you. So with that, I can promise you, John, that I'm very humbled, and I'm beginning my talk with this. I also should, before going any further, I should give credit to Xiong Wei, a Chinese PhD student that essentially showed up at my door in Sweden when I was on sabbatical a year and a half ago. Had a data set, was there for 1 week, and over this week essentially analyzed the entire data set. Unfortunately, he disappeared into Mongolia to set a watershed study, so I have very limited communication with him. And some of my interpretation of the data may be slightly off. But I have an excuse which I'll give you at the end of my talk. Also, I should thank Jean-Christophe Domecq, who is the person who actually ran some of the models, Musica essentially, for testing the effects of some of the hydraulic redistribution that is occurring in forests, and Asko Nurmitz, Chris Soishi, and Sari Palmroth for some of the data that they provided without it being published yet. I was actually quite surprised, and maybe that tells you that I'm really not part of this community, that the term hydraulic redistribution didn't appear once. I really paid attention, and I wasn't going for coffee or to the bathroom while anybody was talking, but I haven't noticed it once. And it's very interesting because when you go to eco-physiologist meeting, that's a term that is actually brought up relatively regularly in order to account for potential positive influences, typically, of that process on the water and carbon budgets of trees and ecosystems, mostly ecosystems, because ecophysiologists really don't look at individuals, they look mostly at a stand level, at stand scale phenomena. But explained based on physiological processes. So for the two of you that might not know what hydraulic redistribution is, eco-hydrologists typically, eco-physiologists typically think of it as the water that passes through roots back to soil. So it's taken up from roots and passed through roots back to the soil. So you could have a system, for example, in which plants dry down the soil during the day. The upper soil is dried more because it's more occupied with roots. And during the night, when water loss is very minimal, typically considered to be as cuticular water loss, not through stomata, you would have an uptake of water from areas with high water potential that then is emitted through the roots back to the soil in areas of low water potential, therefore sort of evening the soil moisture content vertically. There is another process that is discussed occasionally, and that is that large trees typically have deeper roots. They have roots also in shallow layers, so they can use the water in shallow layers with the trees that are smaller and have shallower root systems. But at the same time, at nighttime, when there's no transpiration, they emit water back into the shallow layers, therefore supplying moisture to neighbors that have shallower root systems, and in fact, if you want, supporting the ecosystem in general by redistributing the available water. So in fact, those concepts are so general that if you go to Wikipedia, you can find those two set of processes actually mentioned. One is mentioned as reduced harsh or horizontal heterogeneity. That's typically associated with agricultural unevenness of irrigation. And the second one is increased water availability through this nighttime lift that I described before. And as I said, this is a hot topic in ecophysiology, and this is just a recent synthesis of papers that look at this hydraulic redistribution, and hydraulic redistribution can be just one directional, mostly from the bottom up, but it also can go from wet layers above when you have rain events that deliver backwater down to deeper soils that dry down over longer dry periods. What you can see from this analysis is that in pretty much most systems, modeled, which is the red bars, modeled estimates are at the upper end or above any estimates that is from direct measurements, such as moisture dynamics in the soil, stable isotope based, or reverse flow based with subflux systems that are stuck in roots that are in shallow layers versus deep layers. So it seems like the models sort of catch the upper end of the measurements or slightly above. So what could explain that, and is hydraulic redistribution as important as we actually thought for quite some time? And that is important now because models begin to incorporate hydraulic redistribution into them, at least models that people that are in my community are using. And recently, I think we become more and more aware in the community that nighttime transpiration can actually be fairly substantial, especially if the vapor pressure deficit stays sufficiently high, because it seems like stomata stay open. And much of the data that I will describe to you, and this is mostly going to be a data-based sort of talk, much of the data that I'll describe come from one of several systems that measure the flow of water in stems of trees. And there are the Granier-type sensors that are shown here. I will not go into details because there are several, and all of them have good attributes, and all of them have problems. If you really spend the time learning those systems, you can actually solve most of the problems, probably not all, unless you go to great extent, and then it limits the scope of your study. So the first time we actually noticed that nighttime transpiration can actually occur was when we measured subflux in Taxodium. It started actually somewhere here about a week earlier. We had a hurricane, and after the hurricane, we said, let's see what's the effect of reducing leaf area. We knew that the leaf area dropped by about 35%. Let's see what's the effect of that on transpiration. So we went back and restarted the experiment and looked at the results. But one thing that sort of struck us at the end of the time was that little blip that we saw at night. Now, that is not unusual. We see it very often in our measurements, and that typically is associated with nighttime precipitation occurring after the recharge of the stem is finished. So as night goes in, water uptake continues for a while, then it gets to equilibrium with the soil, it stops, and then the next morning you have flow. However, if you have rains that begin in the middle of the night after that stable point, you can see another pulse goes into the tree, and that's another recharge that occurs as a result of changing in the water potential gradient between the soil and the tree. We were not surprised by that. But this study was in a swamp, and there is no recharge as a result of precipitation. So we looked around and we found out that there was a pulse of warm air that went into the system and went out of the system. And because it went in and went out all during the nighttime, it was clear that it did that. So we wrote a paper about that, and that was very nice. But and we could find only two other papers that saw that sort of a phenomena, and both of them were on very wet mesic systems. So our explanation was wet systems, trees do not care, they keep the stomata open, and thereby they can have the stomata open early morning when the sun comes up, because there is a time lag between the activation of photosynthesis and the activation of the control over the stomata. So they can bypass it and be ready for the light when the light is coming, and they don't have to worry about water because it's a mesic system. Okay, since then, people showed it in most trees that they looked at. In fact, what we see here is yellow poplar, sweet gum, two hickory species and 3 oak species. This is based on subflux. There are about 15,000 points per tree. There are about 40 trees here. So there's quite a lot of data hiding behind those curves that you see. But what it shows is that during the daytime, this is the stomatal conductance that is calculated from subflux. During the nighttime, this is the stomatal conductance that you see calculated based on subflux in relations to vapor pressure deficit. So you can notice a couple of things. And first of all is that the absolute conductance is different, and it tends to be higher in the 2 shade-intolerant species in compared to the shade-tolerant species. You can also see that the nighttime conductance relative to the daytime conductance is higher also in the 2 shade-intolerant species relative to the shade-tolerant species. It's about 40% versus 20% or so. So that is very tempting to start over-interpreting, so I'll do that. And what the point is that one could explain that if plants transpire at night, of course, they limit the hydraulic redistribution. And if you are a shade-intolerant species that grow fast and put deeper root system, there's really no sense for you to supply water to the shade-tolerant competitors that are around you. Okay, that's a good, maybe reasonable explanation. I don't know, we could argue about that. But the fact is that it so happened that those 2 species up here are also diffuse porous species. They have a xylem structure that is very different hydraulically from the 2 species from the 5 or 6 species that we see here, which are ring porous species and have, as I said, very, very different hydraulic characteristics. So it's very possible that what we see here is simply a reflection of hydraulic characteristics differences between those two sets of plants rather than others. Okay, so this information was put, as I said, by JC through a model that is called Musica, where I think JC incorporated the hydraulic redistribution that affects then the gradient between the xylem water potential and the soil water potential, therefore affecting transpiration, and has therefore an impact on GPP. And he ran the model for the stand that I just showed you. And what he found out is that in this warm, moist, deciduous forest, percent of hydraulic redistribution, assuming no nocturnal transpiration, provides about 15% of the annual transpiration of that stand. So 15% of it is brought up from deeper soil to shallower soil. 8%, the number is reduced to about half if you actually account for nocturnal transpiration. So the nighttime loss cuts down. Now, the amount of water that is available, and that, because of the effect on stomatal conductance, it has a similar effect on GPP. So if you are in a business of estimating a canopy photosynthesis, you might want to incorporate a hydraulic redistribution, but if your species or your system have high degree of nighttime transpiration, you actually have to account for that as well. In a preliminary round that JC did on 10 different forests, ranging from wetland-type forest to dry eucalyptus and ponderosa pine forest, the range of hydraulic redistribution not accounting for nocturnal transpiration ranged from 3 to 28%. So if 28% of your model estimate of GPP is dependent on hydraulic redistribution, that's a very important phenomena. However, if half of it is gone because of nighttime transpiration, that's also an important phenomena to consider. Now, forests have, so this is what I'm going to say about the vertical distribution that is a result of hydraulic redistribution. We'll go to spatial, and I heard a lot of discussions, I think, today, and not all of them I totally understand, about the possibility of lateral movement of water, I guess along potential gradient within the soil and how limited it is and so forth. But if you have a root that goes from a dry zone to a wet zone, one could imagine that a lot of water could come out at night if there's no transpiration from the wet zone and be transported into the soil of the dry zone. Pretty much in the same concept as the uplift and downward distribution. Forests, although they do not have the same regular structure that some agricultural fields have, I remember being at Oregon State, a graduate student, and Dave Perry, one of my professors, actually told us to think about forests as essentially like egg cartons, that you have dominant individuals that tend to suppress those around them, and that because they dominate a certain amount of resources, because of their size, they require a certain amount of resources, you sort of will have a distribution that will be fairly regular. Not as regular because there are a lot of other processes that play a role, but fairly regular. And he did some transects in Douglas fir forests and found out that there was a regularity to soil carbon that actually matched the distances between dominant trees. So there was one, he measured other variables as well. So one can imagine that if you put, I'm repeating the same figure, if you put a tree that has this sort of a transpiration rate not far from a tree that has this transpiration rate, both of them are dominant. Root systems is definitely crossing over 15 meters from the stem of a tree. In trees, that's not very unusual. You will have this tree exhausting soil moisture much faster than this tree, and therefore transporting water from here to here, and being able to then utilize it during rainless periods. And in the process, evening up the soil moisture laterally. Now, this evening out is almost, maybe implicitly rather than explicitly, but it's almost essential for a lot of the models that we are running, which are based like 3PG or Forest BGC or Musica, that actually are one-dimensional model and run with an average soil moisture. Because of the nonlinearity between soil moisture and conductance, if this assumption is wrong, your estimates of conductance will be very wrong. And as a result of that, your estimates of GPP will be quite wrong. Okay, so this is a possibility. We didn't study it there. We studied essentially earlier something that, again, I didn't pay attention to. So I'm pretty good at not paying attention to things for a very long time before I realized how important they might be. But we looked at a stand that was incredibly uniform. Actually, if you go to Google Earth, above that stand, you cannot find a stand that is as uniform as this for about 60 miles around the spot. It was selected, therefore, to be the stand that was utilized for the Duke Face site, and before that, for other studies that looked at biosphere-atmosphere exchanges. It was incredibly uniform. It was all cut down at the same date. It was burned. It was planted with the same half-seed genotypes across the entire thing, the same treatment everywhere. And what you see here is something that we saw yesterday, which is quite a large spatial variability of basal area. And when you transfer that to a leaf area index map, you found this sort of conditions. And again, just like we saw yesterday, the lower elevation, which is about 10 meters lower than the upper elevation, the lower elevation had higher leaf area, much higher leaf area than the higher elevation sections. So even the most homogeneous forests are not homogeneous. That's something we need to remember in terms of, at least some of us need to remember because of the way that we model our systems. Now, you look at this area here, and it looks, well, much more homogeneous, relatively speaking. So we look at that, and what we have here is plots that we put around trees. That plots are now 3 meters in radius, so it's not a big plot, all within that blue area. And it has a sapwood area per unit of ground area that ranges about threefold, from 0.01 to 0.03, which is pretty much associated with the same range in leaf area. So on the even more local scale, you find out, again, high variability in leaf area index. And this high variability in leaf area index or competition or site occupancy or whatever you want to call it is translating very nicely to a reduction in the subflux density. And because of the relationship between sapwood area and leaf area, which is relatively invariable, that means a reduction in transpiration per unit of leaf area. Multiplying this by this gives us the total transpiration on that date when soil happened to be fairly wet. And what you see is that the high the areas that were highly occupied by sapwood area, high leaf area, transpired much more than the areas that had lower sapwood area. So you will have high stomatal conductance over here, but because of low leaf area index, you will have lower total transpiration, and therefore you probably will exhaust soil moisture less in that spot than in this spot over here. So, as I was preparing to talk, I really didn't have much to say, so I started digging for some old stuff, and I found out a thesis that was published by Rainer Zimmermann in 1990, never made it into other publications, but in a very German way, he actually measured everything on very little area. So what he has here is, we see here is a cartoon of 3 trees, unfortunately not to size. The distance between the trees is 5 meters and then 3 meters. Okay? The total transpiration is about 3,000 liters for the period for that tree, close to 4,000 liters for that tree, and about 1,000, slightly higher than 1,000 liters for that tree. It was all done with a Chermak type system, for those of you who knows the differences between subflux systems. So it's a rather more accurate system than the Granier, but is more disruptive. And what you see is the transpiration over time from July till October in those 3 trees. And obviously, the small tree transpires per unit of time much less than the 2 large trees. The 2 large trees also show much greater dynamics. They sort of drop in transpiration in both cases. And the drop in transpiration agrees with what we see in the soil. So how was it measured in the soil? Every 50 centimeters about, from between this point and this point, there were soil tensiometers positioned in 25 centimeters and 45 centimeters from the surface, and were measured over the entire period. And so what you see is the buildup of soil moisture tension and then the decay of it as rains begin to come in, days become shorter, transpiration is reduced. But as long as the days are nice and transpiration continues and the rains are not so ample, actually the transpiration of those big trees exceeded the throughfall, and as a result of that, you see this very large buildup of soil water potential. You don't see that under the small tree. You also do not see that those trees which have roots which at least go across, at least, given that they are 15 meter tall, they will easily have roots that expand 7 or 8 meters from the trunk, seem to not affect that intermediate area very much. They might do. We don't do a balance there. We didn't compare, or I didn't compare the tall flux with how much can be supplied at different zones away from the tree. But you can see that the soil moisture potential under the small tree remains actually quite high. Okay. So this is a wet system, and you can say, well, okay, it's a wet system. They probably have enough water for where they are. They don't have to go and get water elsewhere. And so the water just stays there. Sorry, Ignacio. They don't use all of it. There's light effect as well.
3 minutes.
No. 3 minutes?
To the 5 minutes.
Oh, okay. All right. Okay, so I'll speed through it. 2 studies from China. One of them is actually arid. That's in vineyard. The other one in semi-arid. The total annual precipitation here is slightly less than 200, and here slightly more than 600. This is a large forest on a sloping ground, and this is obviously on a fairly even ground. This is the site. The experiment was to measure subflux systems with Kuchera type method. And so those spots represent the plants that were actually measured. The ovals represent places that soil moisture was measured with TDRs. And the dark bars to the left and the right of each vine that was measured represent the meter to 1 to each side where leaf area index was estimated. And one more point, yes. The soil moisture was measured after each irrigation and before the next irrigation. So after a drying cycle and before the next irrigation, next to each one of those vines that subflux was measured with extraction of material and drying it. This is just to show you that there is a very good correlation between subflux scaled conductance versus leaf level scale conductance, but you really need to be careful in how you scale the subflux and the porometry-based conductance. So subflux does give you tons of data, which is really, really nice because you can start parsing the data into small subsets of conditions which are very similar and then express them relative to another variable. So here we have subsets of lights and then related to vapor pressure deficit. If you fit a log linear relationship to that, you actually get a value that we call reference conductance at 1 kilopascal. And then you can look at the reference conductance for each vine in relations to soil moisture when, for example, light is not limiting, temperature is not limiting, etc. Okay, so we see that all of them sort of tend to drop down. All of those 8 vines tend to drop down with water potential, with soil moisture, but they drop down actually at different rates. And when you go and you look at the soil moisture data right next to each one of those vines, you find out that in the 2 drying cycles that we show here, just as an example, you find out that the higher the leaf area, the more of the soil moisture was exhausted from the area right next to that plant. And when you look at the decay rate of the stomatal conductance with respect to the average soil moisture in the vineyard, you find out that it is increasing with a leaf area relative to that of the average. That means that in high leaf area index positions, you actually have a faster drop relative to the average than in low leaf area index conditions, indicating to us that the local leaf area index, very localized leaf area index, dictate mostly the exhaustion of water in that position. The other study I won't have much time to describe, it's the larch. This is the stand. We did the same thing that we did in the study that I described before. We put small plots around each one of the plants. We estimated the sapwood area per unit of ground area or leaf area index in each one of those little plots, and we related that to the same sort of conditions. I won't go through that. That just explains the variability amongst individuals at a given soil moisture or at high soil moisture. And it's all related to light, of course. There's higher competition means you have lower average conductance, therefore lower subflux density. But here is a similar description of the decrease in, this time, subflux density, not conductance, like we saw in the vineyard, with respect to average soil moisture at the site. You can see that all of them decrease with respect to, as the soil moisture became more and more negative. But when we look at the rate of decay, we find out that you can explain, again, the rate of decay with respect to the local leaf area. Leaf area within 3 meters of the tree, the leaf area index within 3 meters of the tree, dictates how quickly the soil moisture was exhausted right next to the tree.
Now 3 minutes without.
Okay. So the point that I was trying to make with all 3 of those examples is that it seems that in very wet system, full canopy forest, a linear system, if you want, with irrigation and very, very dry, and in a large stand which is rather messy in structure and actually intermediate in drought or semi-arid, it seems like the local moisture right next to individuals really reflects the leaf area index or the competitive strength of the system right at that spot. And that doesn't, despite the fact that sometimes you can take and follow a route for 15 meters away from a tree, it seems that those routes that go away from a tree have very little effect on the water economy of those individuals. And so the reflection of that is that, of course, that if you use the average you average those 2 values and you calculate stomatal conductance, you'll get much higher value than if you use each one of those values to calculate stomatal conductance because of this nonlinearity between the two and the effect of that on GPP. We did a very quick model exercise and compared two situations, 1 and 3 meter square leaf area versus 2 meter square leaf area. And ratioing this to that, you find out that soil moisture is lower and the effect on stomatal conductance is actually quite large. And when you average it over the entire period, your average soil moisture estimates are actually 6% lower, and conductance is about 12, so it's going to have a similar effect on GPP. And I know it's inappropriate, but my father wanted me to invite him to this conference, and I know I couldn't, so I decided I'm going to thank him and my grandfather for getting me interested in forests and trees. And I said I'll leave you with some statement that will excuse my performance, at least my lies, and hopefully that'll be useful for all of you. And those of you. Yeah.
Could you go to the your curve where you had the blue lines and the green lines, where you were making the calculation of conductance versus vapor pressure deficit?
Yes.
Yeah, these guys. And so the blue, you calculate that, if I understand, just using the vapor pressure deficit itself, and
Subflow.
And subflow.
and subflow.
And
But-
subflow at nighttime after you correct for the capacitance of the tree.
Okay. And then the green.
The green line is daytime.
Okay.
And the daytime you also correct for the capacitance of the tree. And there are a couple of, there are different ways. Gabi was involved in one of them where you use porous medium approach to estimate capacitance, and then you allow it to go. You have the RC circuit sort of approach with single components or multiple components. There are various ways. People use various methods to deal with capacitance. All of them have some issues. The porous medium is difficult to parameterize.
Okay, last question over there.
Ram, one question, fascinating thing, lecture. If I understood you correctly, the very extended roots do not have a major influence in the soil moisture state. Why does the plant invest so much carbon effort in these long roots, especially in arid systems?
Well, I don't know. I don't have an answer for you. I can speculate. And speculation is that roots are very...
The microphone.
Oh, sorry. Roots are very often thought by peoples depending on their field. So I work mostly with nutritionists, and for them, roots are nutrients. For us, roots are maybe water, but in fact, you have both of those, and so one thing is clear is that you do need the roots for other things as well, and as a low flux actually delivers very similar amount of nutrients that a high flux, because when you have a high flux of water into a plant, typically you find out that the concentration of nutrients in the xylem sap decreases. So you don't care if you have low flux that are coming up from a far distance, from a long distance. You can still take a little water and quite a bit of nutrients coming with it. But that's just one speculation.
Thank you.
Okay, we have to move on, and you have to go to your meeting. So we're just going down the corridor, remaining at Duke, North Carolina.